Welcome to today's webinar from the International Alliance of ALS-MND Associations, our fourth annual Global Clinical Trials Update. For this webinar, we have captions available. If you're joining us live on Zoom, please check the chat box as we are adding the instructions right now, or scan the QR code on screen if you have another device available. Just give me a moment. I'm just going to add these now. If you're watching the recording in our YouTube channel, you will find instructions in the video description on how to activate the subtitles. Para este seminario web tenemos subtítulos en español disponibles. Si se unen a nosotros en vivo vía Zoom, verifiquen en la casilla del chat, ya acabo de agregar las instrucciones en este momento, o pueden acceder a la traducción por medio del código QR en la pantalla usando otro dispositivo. Y si están viendo la grabación en nuestro canal de YouTube, encontrarán las instrucciones en la descripción del video sobre cómo activar los subtítulos. For this webinar, we are using automated captions. Please bear in mind that this kind of translation is not perfect and many variables can affect accuracy, and errors are possible. However, even if a few words are incorrect, you should still be able to get the meaning in context. For those of you who don't know me, my name is Jessica Mabe, and I'm the Programs Coordinator at the International Alliance of ALS-MND Associations. Before we begin, I'd like to take a quick moment to ask for your support in sharing our Fundamental Rights Survey, which will be closing soon. You can access it by scanning the QR code on your screen, and I'll also add the link in the chat shortly. Our goal is to reach 3,000 responses, and the survey is available in 15 languages. It only takes about 15 minutes to complete. If you've been personally affected by ALS-MND, whether you're living with this diagnosis or are a current or former caregiver or a blood relative of someone with the disease, we'd be so grateful if you could share your experience. The survey is open worldwide, so no matter where you live, your input can make a real difference in helping us better understand and improve care and support for people and families affected by ALS-MND. Every response truly counts, and thank you so much for your support with the survey. Now, let's get started. Today, we will be getting information on ALS-MND treatments currently on phase 3 or soon to start phase 3 clinical trials. Today, we are joined by Biogen, Clean Nanomedicine, Ferrer and Prelenia, Ionis, the Iran University of Medical Studies, and the Mario Negri Institute of Pharmacological Research. Thank you all for joining us today to be able to give these updates to our ALS-MND community. Our moderators today are Dr. Nadia Sethi and Dr. Martina Majo. Dr. Nadia Sethi is co-chair of the Northeast ALS Consortium Patient Education and Advocacy Committee. A dentist by training, she transitioned to research-focused ALS-MND advocacy after her husband's diagnosis with the disease in 2019. She serves on the programmatic panel for the Department of Defense's ALS Research Program, the Scientific Advisory Council for the International Alliance of ALS-MND Associations, and the Leadership Board of Genetic ALS and FTD and the Legacy. She also manages a social media community of over 7,000 focused on sharing scientific content with the patient community. Previously, she served as Director of Community Outreach at ALS TDI. Dr. Martina Majo joins Nadia moderating today's webinar. Dr. Majo has extensive experience researching disease mechanisms of ALS-MND and frontotemporal dementia. Dr. Majo earned her bachelor's in biotechnology and master's in pharmaceutical biotechnology with honors from the Sapienza University of Rome. She then received a PhD in clinical neuroscience from King's College London and completed her postdoctoral training at University of California in San Francisco. In addition to her academic training, Dr. Majo worked as a principal investigator in the industry sector, directing several National Institutes of Health projects around ALS-FTD in vitro modeling. She joined the Alliance as Scientific Director in 2024 and has been coordinating the Alliance research portfolio. Now, some final instructions. Any questions you have, please write them in the Zoom Q&A box. I also want to take this opportunity to thank everyone who has pre-submitted their questions. There is a lot to cover, and we will do our best to answer as many questions as we can. I will now turn it over to Nadia and Martina, our moderators for today.
Thank you, Jess. That was wonderful. Thank you, everybody, for connecting, or whoever's watching this recording. We're very excited about this webinar, and we are going to start with a presentation from Biogen by Steve Garofalo.
All right, so many thanks for the opportunity to provide an update on the tofersen program. My name is Steve Garofalo, and I'm a medical director at Biogen, and I'm very grateful for your time today. So we'll first start with a regulatory update on tofersen, which has been approved in 41 countries. These include the US, EU, China, Japan, Canada, the UK, as well as additional geographies. Notably, Biogen continues to advance filings in additional regions where the totality of evidence is currently being evaluated by global health authorities. So as you may know, in approximately 2% of all people living with ALS, the disease is due to a mutation in the gene encoding the superoxide dismutase 1 or SOD1 enzyme. Current evidence suggests that this confers a toxic gain of function mechanism, which then leads to accumulation of a toxic form of the SOD1 protein that results in motor neuron degeneration. Tofersen, which is shown in green, this is an antisense oligonucleotide. It binds to SOD1 messenger RNA, which then targets the transcript for degradation via the RNase H enzyme. Reduction of SOD1 messenger RNA then leads to reduction of SOD1 protein levels, which would be expected to slow the neurodegenerative process in SOD1 ALS. And so in this way, tofersen has been designed to address the fundamental disease pathobiology of SOD1 ALS. In terms of clinical development, tofersen has been evaluated in multiple clinical trials. These include the 233 AS101 or Study 101, which consisted of 3 parts, including a single ascending dose phase known as Part A, a multiple ascending dose phase known as Part B, as well as a phase 3 randomized, double-blind, placebo-controlled phase known as VALOR. Following completion of Study 101, participants could enroll in Study 102, which was an open-label extension study where all participants received tofersen. In addition to the completed trials, Study 101 and 102, the ATLAS study, which evaluates the impact of initiating tofersen in pre-symptomatic at-risk carriers of a SOD1 mutation, is currently ongoing. In terms of the current status of the open-label extension study, this trial is now complete with the last participant study visit in August of 2024. Final results from the integrated analysis of VALOR and the open-label extension studies have been presented at multiple congresses this year, including NCALS and NEALS, with a manuscript that is currently in development. And overall, these results demonstrated that tofersen was associated with reductions of decline and measures of clinical function, respiratory function, and muscle strength than would be expected based on the disease natural history, with earlier treatment initiation associated with reduced decline on these measures, as well as a greater proportion of participants who exhibited improvement from baseline on these measures, as well as an extension of event-free survival outcomes. And so these data help us better understand the long-term effects of tofersen in people living with symptomatic SOD1 ALS. So now shifting over to ATLAS, which as mentioned is designed to evaluate whether initiating tofersen pre-symptomatically in at-risk carriers of a SOD1 mutation upon a neurofilament elevation will delay the time to emergence of ALS and or slow the decline in function after disease onset. Notably, this is the first interventional study in pre-symptomatic ALS that is grounded in the seminal findings from the pre-symptomatic familial ALS study, which first identified elevated neurofilament levels in individuals who were at genetic risk of developing ALS who later developed the disease. ATLAS is a 4-part study. Part A is a natural history run-in in which neurofilament levels are monitored on a monthly basis, and this part includes pre-symptomatic adult participants with a confirmed SOD1 mutation. Participants in Part A, whose neurofilament levels meet the pre-specified threshold, which is 44 picograms per mil, as well as a change from Part A baseline of 10 picograms per mil or greater, will be screened for Part B, assuming no alternative non-ALS cause for the neurofilament elevation has been identified. Part B is a randomized, double-blind, placebo-controlled study part where participants will receive either tofersen or placebo via intrathecal injection. Participants in Part B who meet the criteria for clinically manifest ALS can then be screened for Part C, which is an open-label extension where all participants receive tofersen. And then Part D includes eligible participants from Part A who meet the criteria for clinically manifest ALS prior to a detectable and confirmed neurofilament elevation. This is an open-label study part where all participants receive tofersen. This slide highlights some of the key eligibility criteria for participation in the ATLAS study. Of note, this is only a subset of the eligibility criteria, and individuals need to meet all study eligibility criteria in order to participate. So for Part A, these are individuals who are 18 years of age or older, are clinically pre-symptomatic, have a plasma neurofilament light chain level below the study threshold, and then have a confirmed SOD1 mutation that is associated with higher complete penetrance and rapid disease progression, as defined by a mutation in the protocol or as adjudicated positively by an independent mutation adjudication committee. For Part B, eligibility includes a neurofilament level above threshold, clinically pre-symptomatic, and then women of childbearing potential must practice effective contraception during the study and for 5 months after their last dose. In terms of ATLAS study status, enrollment in Part A is now complete with 158 participants enrolled. Enrollment continues in Part B with a target enrollment of 28 individuals, and enrollment in Part C and D is also ongoing. The study enrolled Part A participants at 27 sites across the 12 countries shown here. Finally, and most importantly, these studies would not be possible without the extraordinary efforts of study participants, their caregivers and families, steering committee members, investigators, site staff, and the wider community. It's only because of this collaboration that we can evaluate the safety and effectiveness of investigational treatments with the goal of developing therapies that can make a real difference in people's lives. So thank you so much for your time today.
Thank you so much, Steve. And next up we have Benjamin Greenberg from Clean.
Hello, my name is Dr. Benjamin Greenberg, and I serve as the head of medical for Clean Nanomedicine, and I'm happy to be with you today. Today's presentation will include a number of forward-looking statements. ALS is a complex neurologic disorder with multiple different pathologic processes that have been implicated in the death of neurons. This includes the toxic aggregation of proteins, oxidative stress, glial activation, inflammation, and hyperexcitability. Regardless of which of these pathologies play a role in the death of neurons, multiple studies have implicated mitochondrial dysfunction as a common end pathway of these insults related to NAD deficiency and ultimately motor neuron death and the symptoms of disability and death that our patients suffer from. What is CNM-Au8? This is a nanocrystal suspension of gold, orally administered, targeting energy metabolism and oxidative stress. It's able to cross the blood-brain barrier and ultimately able to enter neurons and mitochondria themselves. Within the mitochondria, the unique structure of these gold atoms allows it to catalyze NADH to NAD, thereby correcting for the energy deficiency and mitochondrial dysfunction found within ALS patients and ultimately leading to prolonged neuronal survival. In clinical trials, CNM-Au8 has been studied across over 1,000 patient years, with the longest patient having a duration of exposure greater than 6 years. Treatment-emergent adverse events have been mild to moderate, and there have been no drug-related serious adverse events. This clinical trial portfolio has included 2 double-blind, placebo-controlled phase 2 clinical trials in ALS, the Rescue ALS and Healy ALS platform trials, as well as a number of expanded access protocols. Ultimately, across these clinical trials, we have found CNM-Au8 improve survival, delay time to clinical worsening, and have an impact on critical biomarkers of disease. Let's focus on neurofilament light chain as a key biomarker of disease progression in ALS. NFL is a cytoskeletal protein that provides structure and support for the cell and is highly specific for neurons. Axonal damage leads to release of NFL and an increase in levels in both the spinal fluid and in the blood. And indeed, patients with a high NFL at baseline are predicted to have a faster and more severe progression in the setting of ALS. When we look to clinical trials where CNM-Au8 was compared to a placebo, what we find is NFL rises as expected in the placebo arm, but fails to rise in individuals treated with CNM-Au8. Indeed, after an open-label extension, individuals who transition from placebo to CNM-Au8 treatment see a relative decline in their plasma NFL levels over time. So what is the evidence linking plasma NFL and the CNM-Au8 clinical benefit? We can look at this both at baseline levels and at decline over 24 weeks, linking to a difference in time to death or permanent assisted ventilation through month 12. First, let's look at individuals based on their baseline NFL characteristics. In the Healy trial, if you look at plasma NFL levels at baseline, either as greater than the median, as shown on the right, or the upper NFL tertile, as shown on the left, we see a greater than 80% reduction in death for individuals treated with CNM-Au8 as compared to placebo. Additionally, if you ask the question, who was more likely to have a decline in NFL during the double-blind period, we find that individuals treated with CNM-Au8 have 57% of individuals having a decline in NFL, compared to only 34% of those treated with placebo. Linking the long-term survival benefit in participants with any NFL decline at 24 weeks, we find that overwhelmingly those individuals treated with NFL had a lower risk of death or permanent assisted ventilation based on a decline in NFL. The time to death or permanent assisted ventilation was most pronounced in individuals who experienced a decline in NFL, and this was more common in individuals treated with CNM-Au8. All of this phase 2 clinical trial data has led to the planning of a global phase 3 trial, the Restore ALS trial. This trial of CNM-Au8 in a double-blind placebo-controlled fashion will allow us to establish the long-term safety and efficacy of this agent. The objective is to investigate the effects of CNM-Au8 on survival and a delay in clinical worsening events for ALS patients. Participant criteria is listed here, and the overall trial design is listed below. Importantly, the primary endpoint of this trial will include survival, while lead secondary endpoints include survival or permanent assisted ventilation, and then a composite rank hierarchy looking at a variety of clinically relevant outcomes. Other key secondary and exploratory endpoints are listed here. Initial launch milestones include the FDA and EMA alignment on study design and endpoint selection, submission and approval by the Sydney Health District, and as well as clinical trial notification submissions and lead sites in Australia being identified, as well as having regional governance submission under review. CNM priorities and next steps are listed here, including continuing regulatory agency collaboration, which is ongoing, a global phase 3 initiation, which is expected in Q1 of 26, and collecting and reporting of long-term ALS experience through our multiple EAP programs. If you have any questions, we encourage you to reach out, and we are happy to engage with you. Thank you for your time, and happy to take your questions.
Wonderful. Thank you so much, Benjamin. Next up, we have a joint trial by Ferrer and Prolenia, which is the Prevail trial, and Luby Alvarez will be presenting.
Hello, my name is Lubia Alvarez. I am Clinical Development Medical Lead at Ferrer, and we are very excited to announce the PREVAIL study. This is a phase 3 randomized double-blind placebo-controlled study evaluating efficacy and safety of predopidine in participants with ALS. The study is being co-sponsored by Prelenia and Ferrer, and is being conducted in collaboration with both NILS and TriCulture organizations. Some disclosures before we start. Predopidine is an investigational drug in clinical development for ALS, which is taken orally twice daily. The drug binds and activates the sigma-1 receptor, which is known to promote healthy neuronal cell function and survival through enhancement of multiple key neuroprotective pathways. A phase 2 clinical trial was conducted through the Healy's ALS platform study, and while the primary and secondary endpoints in the entire population did not show benefit compared to placebo, benefits were observed in post-hoc analysis in a population of early and rapidly declining participants with significant slowing of progression, measured by ALS-FRS-R total score, respiratory and bulbar subdomains, and quantitative speech characteristics, while maintaining a favorable safety and tolerability profile. Based on these results, Prelenia and Ferrer will initiate the PREVAIL phase 3 study beginning in early 2026. The study will enroll 500 participants, randomized in a 3:2 ratio to predopidine 45 milligrams twice daily and placebo plus a standard of care, for a 48-week double-blind placebo control period. Following this initial double-blind period, all participants will receive predopidine for an additional 48-week period as an open-label extension phase. The study will enroll an early-onset, rapidly progressing population of adult participants with ALS. The primary endpoint will be ALS-FRS-R in week 48, with secondary endpoints measuring overall survival, changes in speech, lung function, and quality of life. The study schedule of assessments will include 8 clinic visits and 5 phone calls throughout the 48-week placebo control period. We plan to initiate enrollment at approximately 20 sites in Canada and the US and 35 sites across Europe. The first participant enroll is currently targeted for Q1 2026 in the US, and we expect approximately 1 year of study recruitment, with a primary readout targeted for the first half of 2028. We will support our study participants with an open-label extension where all of them will receive predopidine for an additional 48 weeks, as well as by covering expenses for study visits such as transportation, meals, and accommodation. It is important to note that at present, predopidine is only accessible through participation in the clinical trial. If you have any questions following this presentation, please feel free to reach out to Prelenia or Ferrer via the contact information listed here. Also, stay tuned for updates in clinicaltrials.gov and clinicaltrialsregistered.eu, as well as on our upcoming website, www.prevailstrial.com. Finally, we want to thank you for your interest in PREVAIL study.
Thank you so much, Lubia. And next up, we have Stefan Miller from Ionis.
Hi, I'm Stefan Miller from Ionis Pharmaceuticals. Later on, we'll be joined by Rob Goldstein-Mahone from our patient advocacy group for the Q&A session. So we both would like to thank the Alliance for giving us this opportunity to give you an update on the Fusion study. This is a fully enrolled phase 1 to 3 study of ulefnersen, also known as ION 363, for people living with FUS ALS. Here are our disclosures. We are both employees and shareholders of Ionis Pharmaceuticals. So the outline of my talk is that it will be What is FUS-ALS? What is the drug ulefnersen? And then finally, how are we testing ulefnersen as a potential therapy for FUS-ALS? So let's start first with what is FUS-ALS. FUS-ALS is ALS associated with mutations in the FUS gene. It's very rare, approximately 1% of all ALS. Mutations in FUS can cause both familial and sporadic cases of ALS. It varies widely in the symptoms at onset and the age of onset. The onset's generally earlier than in the overall ALS population, but it's highly variable, with the age of onsets ranging from as young as 11 years old to 80 years old. I said 11 years old, and it has been reported in very young patients, and that's perhaps the most unusual thing about FUS-ALS. It's very rare in young patients, but it is the most common the most common ALS causing mutation in juvenile and pediatric ALS, although most patients are adults. The overwhelming majority of patients are adults, but when it does occur in young people, it often has a fast progression. So what is FUS? FUS stands for fused in sarcoma, and that it is an RNA binding protein with roles in transcription, RNA splicing, transport, stability, and translation. So very important protein for getting the code in DNA turned into RNA and then the proteins that are necessary for normal function. So what is ulefnersen? So this is a cartoon of its mechanism of action. And showing here in the blue is DNA. DNA is a double-stranded molecule, carries the code for all genes. And people with FUS-ALS have a mutation in their DNA that's shown in that little red bar. That mutation will be carried over to RNA on the way to making protein, and then instead of a normal protein being made, protein is made that is a mutant protein and has a tendency to aggregate or clump in motor neurons, and that is somehow toxic to the motor neurons. So ulefnersen, shown here in purple, is a drug that's part of a class of drugs called antisense oligonucleotides. And ulefnersen binds to that mutant RNA, causing it to be broken down so that the mutant FUS protein is not produced. So ulefnersen reduces the production of that toxic protein, and then the therapeutic hypothesis is that degrading that mutant RNA will reduce the aggregation of the FUS protein, and that will reduce motor neuron degeneration, which will slow or maybe even stop the progression of the disease. Here's the study design. It's a phase 1-3 study with a 72-week placebo-controlled double-blind treatment period. So 72 weeks, almost 17 months. And participants are randomized at the beginning 2-to-1, ulefnersen or placebo. So 2 patients to ulefnersen to 1 patient on placebo. After completion of the randomized period, there's an open label treatment where everyone is able to receive ulefnersen, and will be available for up to 5 years or until the drug becomes commercially available. The study drug, ulefnersen or placebo, is administered via lumbar puncture or spinal tap. That's every 12 weeks with an additional loading dose after the first 4 weeks. And then the assessments in the study are very common across other ALS studies. There is the ALS-FRS, which is the functional rating scale, measurements of breathing capacity and muscle strength, and other questionnaires to measure physical function and quality of life. There are many safety assessments. There's MRI, ECG, safety labs of blood, urine, and cerebrospinal fluid, and of course all adverse events are carefully monitored. Here are some of the key eligibility criteria. Of course, an approved mutation in the FUS gene. Patients as young as 10 years of age can participate, and they have to, of course, have signs or symptoms of ALS, and the patient cannot already have a tracheostomy or require ventilation of more than 22 hours a day. Background therapies like riluzole are not allowed, and tofersen, the drug for SOD1 ALS, is not allowed. As I mentioned, the study is fully enrolled, and I don't have it on the slides, but the total study population is 89 participants. The study enrolled these participants at 20 sites in 14 countries, so mostly US and Europe, but also 3 countries in Asia, South Korea, Japan, and Taiwan. Enrollment's was completed in July 2025, and enrolled patients are ongoing and receiving their dosing, and we're collecting all of those assessments. So very exciting time for the study, and we're really looking forward to getting results on this study, which should be at the end of next year. Another great thing that happened in addition to full enrollment this past year is that Ionis signed a licensing agreement with Otsuka Pharmaceutical, and Otsuka Pharmaceutical will be responsible for the commercialization of ulefnersen if this study is successful. So this is great news for the program and great news for FUS-ALS, because Otsuka is, like Ionis, very committed to bringing transformative medicines to patients in indications with high unmet need. So here's where you can find more information about the study on clinicaltrials.gov. Again, I'd like to thank the Alliance for the opportunity to talk about this study, and we are deeply indebted to the people participating in the trial and their families, and also to the Fusion investigators and site staff that make this study possible.
Well, thank you so much, Stefan. Next up, we'll have Homa Talebi from Iran University of Medical Studies.
Hello, my name is Homa Talebi, and I'm a PhD candidate in neuroscience at Iran University of Medical Sciences. It's a great honor to be part of the 4th International ALS webinar in Toronto, and I'm truly grateful for the opportunity to share our ongoing work with this global community. In this brief presentation, I'll be discussing the clinical trial currently underway at our center, which focuses on evaluating the efficacy and safety of low-level laser therapy on motor and non-motor symptoms in patients diagnosed with amyotrophic lateral sclerosis. ALS remains one of the most challenging neurodegenerative diseases, and every step toward understanding its mechanism and identifying potential treatment brings us closer to meaningful change for patients and their families. I'll begin with a quick overview of the study. As you know, ALS remains a devastating neurodegenerative condition with limited treatment options. Photobiomodulation has shown promising neuroprotective and anti-inflammatory effects in a preclinical study, and our trial aims to evaluate its clinical potential in a controlled setting. Photobiomodulation involves the application of low-level red or near-infrared light to body tissue, where it is absorbed by mitochondrial chromophore, particularly cytochrome C oxidase. This interaction enhances mitochondrial respiration and ATP production, leading to improved cellular energy metabolism. Additionally, photobiomodulation modulates ROS level and promotes the release of anti-inflammatory cytokines while reducing pro-inflammatory mediators such as TNF-alpha and IL-6. These effects collectively contribute to neuroprotection, reduce oxidative stress, and potentially slower disease progression in ALS patients. This is a single-center, randomized, double-blind, sham-controlled trial involving 34 eligible ALS patients. Participants are randomly assigned to one of 2 groups. The intervention group receives active laser therapy in addition to standard care. The control group undergoes sham laser procedure alongside standard care. Randomization is computer generated and managed by an independent epidemiologist. The study consists of 30 treatment sessions over a 6-week period with 5 sessions per week, each lasting 20 minutes. Clinical and biochemical assessment are scheduled at the 3 key time points: baseline, after session 15, and after session 13. We are evaluating range of outcome measure. Clinical outcome include the ALS Functional Rating Scale Revised, ALSFRS-R, and the Motor Unit Number Index. Biochemical markers include inflammatory cytokines such as IL-6, IL-10, and TNF-alpha, as well as oxidative stress indicator like malondialdehyde and superoxide dismutase, and light chain neurofilament. Patient report outcome cover quality of life, pseudobulbar effects, and sleep quality. All adverse events are closely monitored and documented. Based on prior study, photobiomodulation is considered safe and well tolerated with minimal side effect. The clinical trial is underway at the Neurology Department of Firuzgar Hospital, a major academic medical center located in Tehran. Our primary focus is on the clinical trial investigating photobiomodulation for ALS. We are not offering a potential program at this time. Thank you for your attention, and I look forward to future collaboration and discussing with fellow researchers in this field. Thank you so much.
Thank you so much, Homa. And next up, we have Elisabetta Pupillo from the Mario Negri Institute for Pharmacological Research.
Good morning. Today I will update you on a randomized phase 2/3 trial on the biological and clinical effects of acetyl-L-carnitine in ELS. The project received financial support by the FightMND, an Australian association, and the sponsors are the University of Sydney in Australia and the Mario Negri Institute for Pharmacological Research in Milan, Italy. Previous preclinical studies and a randomized trial and an observational study in ELS subject with mild functional impairment at entry support the putative efficacy, safety, and tolerability of acetyl-L-carnitine, also called ALCAR, when associated to riluzole compared to riluzole alone. Although promising, the study results are insufficient to support the claim of efficacy for ALCAR. First of all, because the clinical effects of the drug were not accompanied by the investigation of biological markers of the pathophysiology of the disease. Secondly, no study verified an adequate dosage to be used in ELS. Based on this, a randomized phase 3 clinical trial is needed to document the effect of the treatment on some markers of the disease mechanism and the efficacy of 2 different dosages. So what we are going to do, we are going to perform a phase 2/3 multicenter randomized double-blind placebo-controlled trial on acetyl-L-carnitine in subjects living with amyotrophic lateral sclerosis. The primary study aim is to assessing the efficacy of ALCAR at 2 different dosages. So we will test 1.5 grams per day and 3 grams per day on the progression on functional disability as measured by the ELS-FRS-R scale. Then we have a list of secondary study aims, clinical and biological. And also we will test the tolerability and the safety of the drug by identifying unexpected adverse events. 246 subjects will be enrolled in 1 Australian site and 18 Italian ELS sites. Here is the inclusion and exclusion criteria that they are the most common inclusion and exclusion criteria. Each subject will be randomized to receive the 1.5 or 3 grams of acetyl-L-carnitine or placebo for 48 consecutive weeks. We have a permuted block randomization scheme, 1 to 1 to 1. After enrollment, each participant will be followed up until death. The visit assessment will be performed at randomization, then at week 4, 12, 24, 36, and 48. A general assessment and neurological examination will be performed in addition to blood sample collection to test the biomarkers, and the functional assessment measured by the ELS-FRS air scale and the spirometry, then the ICAS scale, a quality of life scale, so the ALSA-Q40, and the drug compliance. Here is the list of all sites that are involved in this trial, so Italian ELS Center and one center located in Australia in Sydney. Here I would like to show you how the sites are located in Italy. So we have the representation of most area of Italy from north to south of Italy. The subject enrollment started in May 2025 and the study will end in June 2027. Today we have enrolled 28 subjects and all of them are on treatment. I would like to thank you for the attention and also I would like to thank you all the investigators and all subjects living with ELS that are enrolled or will be enrolled in this study.
Sorry, that was wonderful, Elisabetta. Thank you. So now we're gonna enter the Q&A session, and we'll have Steve Garofalo from Biogen joining us, Marjan Spassi from Clean. For the Prevails trial for Rare and Prolenia, we have Diderik Boot and Luby Alvarez, both on the call. And we have Elisabetta Popillo from the Mario Negri Institute. Welcome, everybody. You can turn your camera on and we can start with questions. Just letting everybody connect. Steve, given that you were the fastest at coming online, I wonder if you could answer. We had many questions, so I don't know if we can get to all of them, but we had a question specific to SOD1 and tofersen, which is about the well, the question is regarding the possibility that SOD1 proteins may exist and be overexpressed in sporadic patients. Should tofersen be tried to in sporadic ALS?
Yeah, thank you so much for the question. That's an excellent question, and it seems to be getting some traction, especially in the academic community. And I've seen some presentations at Niels and other conferences relating to this. There is some evidence, primarily from post-mortem autopsy studies, showing evidence for SOD1 pathology, so accumulation of disordered, misfolded SOD1 in patients that do not have SOD1 ALS or a SOD1 mutation, so other familial forms of ALS or sporadic forms of ALS. So there's been a number of studies that have evaluated this. Some studies show supporting evidence, and then some studies do not replicate these findings. So at the moment, there seems to be some level of uncertainty about whether there is the presence of SOD1 pathology in non-SOD1 forms of ALS. And then also, if SOD1 pathology is present in non-SOD1 forms of ALS, how might it contribute to the disease mechanisms of ALS? So I think right now there's a bit of uncertainty about that, but there certainly is interest in continuing to explore that hypothesis. So that's what I would say at this point. Excellent question.
Thank you.
Thank you so much.
Thank you, Steve. And I'm going to pick up the next question here. This is for all of the panelists. How are your trials accounting for heterogeneity? We can start with whoever's willing to go first.
I can start. I think for CNMA U8, our goal is to, and what we believe the efficacy of CNMA U8 would be a heterogeneous population. And so for us, we encompass that by having the baseline NFL score as part of the inclusion criteria, as well as the TRICALS risk score range. What's most important for us for CNMA U8 is to make sure that we have enough time on therapy. We've learned through our preclinical work and our phase 2 programs that you really need at least 3 months on therapy to reach steady state and then ultimately have those clinical effects. So for us, you know, that range of having folks that are not too fast of progressors or too slow is quite important. But other than that, we believe that CNMA U8 can actually be quite helpful on top of standard of care for a wide range of patients.
Maybe I can brief.
Yeah, go ahead.
No, go ahead, Lubia. Go ahead.
Yeah, no, so from our side, of course, we're also specifying inclusion criteria, including patients with early onset of the disease. We're also using the TRICALS risk assessment, and of course, also everything that is related to the ALSFRS score that is a specific of specific scores in subdomains as bulbar domains and respiratory the subdomains. This will also help us build this population, which we have evidence that will have more effect with our treatment. Did you want to add anything, Didrik?
Yeah, maybe just to add briefly that what we're trying to do is to, let's say, replicate the positive results we saw in the phase 2 trial. So we saw an effect in a subgroup of patients, and those criteria are then used to select the patient population for the upcoming trial. So that's our main goal. So we have similar criteria to focus and try to get a somewhat homogeneous participant population.
Please, Steve, go ahead.
Oh, sure. I'm open to others if you also want to comment. I know I answered the first question, so I can pause if others want to jump in. Okay, I was just going to say for Atlas, this study is enrolling, as mentioned, individuals that carry a SOD1 mutation that is associated with higher complete penetrance and rapid disease progression. And so in terms of heterogeneity, we know that there is a fair amount of heterogeneity in SOD1 ALS across different mutations, but we've included that criteria to try to have as homogeneous a population as possible. And we know from the results from the pre-symptomatic familial ALS study, these were primarily in individuals that carried a SOD1 A5V mutation, which is associated with very rapid disease progression. It's a very well-characterized mutation. And so there is some level of uncertainty since we are including additional mutations within the context of the study. But we thought it was important to broaden the scope of the mutation selection to more generalize the results across the SOD1 ALS population. So while there is some uncertainty related to mutation type, we have a number of ways that we've tried to address that in this study. And so we have several stratification factors for randomization. One includes whether individuals have an A5V mutation or not. So individuals will be randomized according to that stratification, as well as the level of neurofilament prior to randomization in Part B, as well as age as well, individuals on the basis of whether they are 30 years of age or older, which reflects the population that neurofilament levels were identified to be elevated in individuals that carried a SOD1A5E mutation in the presymptomatic familial ALS study. So we have a few different ways to try to control for disease heterogeneity, in addition to having the list of predefined mutations in the ATLAS protocol, as well as a mutation adjudication committee, which independently evaluates all SOD1 mutations for inclusion within the study based on family pedigree information, information in the scientific literature or other databases. But it's a really excellent question because we know that overall trial results can certainly be influenced by study population heterogeneity, and that's really important to control for. So thank you so much for the question and great answers from the panelists.
And Elisabetta, you have your hand up.
Yes, thank you for the question. I think that it's very difficult to define the heterogeneity of ELS because there's a lot of characteristics that play a role in the disease classification and also in the heterogeneity. To manage this, we decided to enroll subjects in the early stage of the disease with a small impact on the respiratory function and also in motor function in order to be able to evaluate if in the early disease stage, fast and slow subject will have the similar effect, if possible. So if we will have a good power, we will perform additional exploratory statistical analysis in order to evaluate if there's some difference in some subgroup. For example, bulbar versus spinal onset, or fast and slow progressors, or based on the disease duration or something else. But unfortunately, we are not able to manage each kind of heterogeneity.
Thank you so much. That was really well explained from all of you. Unless there are other comments on this question, we can move on to the next question. Let's give you a few seconds to react if you want to. Okay, so a question for everybody here is, at what stage of ALS would your interventions work best?
Well, in our case,
Anybody
as we just mentioned, since we are already basing our trial in a previous phase 2 trial, we are going for the same, for the population we saw effects in, which is an early onset symptom population with definite or probable ALS defined by ALS Curial classification. So that's where we're going.
I think maybe I can tag on to that because ours is similar. So again, it would be early onset symptomatic patients for CNMAU8. We've also shown in the Healy platform trial, based on our experience, that the earlier, the better to start CNMAU8. And when we compare those that transition to long-term extension, i.e. for the first 6 months in the Healy platform trial, if you remember, the double-blind period was 6 months. And when they transition over to taking active product, they don't ever catch up. So that difference continues to be significant between the groups, although they're now both taking CNMAU8. So our preference and belief for CNMAU8 is also to be as soon as possible.
Yeah, please, Steve, go ahead.
Say that I agree.
Thank you, Elizabeth and Steve.
Yeah, I mean, just great responses. Just to add on, you know, we know the majority of motor neuron degeneration occurs prior to the manifestation of clinical symptoms. So I think it's, you know, treating as early as possible. We've seen this be successful for other disease areas, notably spinal muscular atrophy, where intervention pre-symptomatically really dramatically alters disease trajectories. ALS, of course, is a very different disease, but the concept of treating as early as possible prior to evidence for motor neurodegeneration would certainly be ideal. So, and that's, you know, something that the Atlas study is trying to investigate, whether initiating treatment early in pre-symptomatic carriers of a SOD1 mutation upon a neurofilament elevation can delay the emergence of clinically manifest ALS or reduce the decline in function once ALS emerges. So we think that's really important to evaluate. And it will be very important to continue to identify biomarkers of early disease. So we know neurofilament levels rise in individuals who are at genetic risk of developing ALS. We've seen that validated in other studies for individuals with non-genetic forms. But how can we identify individuals who are likely to develop ALS with a more specific marker so that we could intervene earlier? So I think efforts in the field to move us towards that position will be really helpful as we think about earlier treatment intervention. So really excellent question and great responses again.
Please, Elizabeth.
Sorry, just to add a quick find. I think that it is important to develop or discover a drug in subjects that are in early phase of the disease, not because we are not interested in the other part of the subject, but because if we are able to find a proper drug in those subjects that has a best response to our drug, maybe we will be able also to treat the subject that has a more worse disease. So it is not because we don't want to enroll subjects that are not in the early phase, but because the science needs a very rigorous methodology, and we would like to really find something, but it's very difficult in this very heterogeneity situation.
Thank you.
Thank you so much. I'm going to pick up the next question here. Patients would rather have survival and other clinical measures as primary endpoints alongside the ALS-FRSR score instead of the score alone, because sometimes this fails to capture subtle changes. What's your take on that? And how do we harmonize this in different trials? That was the question submitted. I would love to hear if you're using any novel endpoints also, or if there's any digital outcomes that are being included in your trial. Would love to hear from all of you. Diederik, you have your hand up.
Yeah, I can go first. So, I mean, it's a very good question, very good comment, but ultimately you need to select one primary endpoint. There's no way around it. And the ALS-FRSR is most widely used, accepted by regulators, and a validated scale. So often that one is chosen first. The same goes for our trial. But of course, we do want to measure the effect of our study drug on other outcomes and more subtle outcomes. So for example, we are also doing speech recordings in our clinical trial, which was somewhat novel, but we have an automated speech battery to record the effect of the study drug on speech deterioration in participants with ALS. And on top of that, we have several questionnaires related to quality of life, of course, but again, also to speech. So we're trying to mix up known outcome measures that are generally widely accepted with novel outcome measures that can really show where the effect of our drug lies.
Thank you for that, Marjan.
Yeah, I think I would agree with Steve. For us, with CNMA U8, I think as Diedrich and mentioned, you have to choose one endpoint, and really putting your eggs in the basket of what you saw in the phase 2 program was really important. So just a couple things to add on. For us, the survival benefit was significant as part of the platform trial. So yes, we agree that survival is a primary endpoint that made most sense for us to move forward, although we are looking at ALS-FRSR as well. And you know, we continue to work on ALS-FRSR. I'm aware that Angela Gange is leading a consortium of folks to continue to improve and work upon ALS-FRSR. So although not perfect, it is an outpoint that we continue to use, and we will be using it as a secondary. But, you know, putting your eggs in the correct basket as you move to phase 3 from what you saw in phase 2 is probably the best bet and what we anticipate moving forward with for our program. I would like to say that, yeah, in terms of design of the phase 3, when we initiated the protocol development, we did have a survey amongst those living with ALS, and survival by and large was really the majority outcome selected by those who participated in the survey. So it is, agree completely, it is something that's very, very important.
Thanks, Marjan. And Steve, you have your hand up.
Oh yeah, I was going to say, I agree with what's been said. Great insights. And survival, absolutely critically important when we think about mortality rates in ALS. I think one important consideration is designing a study of sufficient duration, and really knowing the temporal relationship between pharmacokinetics to impact on drug target, to then impacting other PD markers like neurofilament, which give you evidence for impact on axonal injury and neurodegeneration. And then what are the timelines from that to impacting clinical outcomes? So ensuring that there is sufficient duration within the context of a study to evaluate efficacy endpoints as set up based on certain time points. So survival, absolutely important. What is the right time point to measure that when we might see the most impact over the course of a phase 3 study? So that's something that we've learned from within the tofersen program, and just something that we will continue to consider as an important impact on overall trial designs, just making sure that we have the right duration to measure that.
Sure. Steve, I think that was a big learning lesson for us out of the Healy platform trial. We realized, one, that it takes us quite a while for CNMA U8 to reach that steady state concentration, and then to have that clinical efficacy start to show. The double-blind period ended in 6 months. So that taught us, for CNMA U8 in particular, that we need a much longer study to be able to see those benefits that we started to see as we followed patients beyond the long-term extension and even looking at expanded access programs. So completely agree. And that's why the phase 3 program for us is event-driven and will be quite a long program.
Yeah, that's a great point. And just one thought that came to mind too is how do we limit the time on placebo for potentially efficacious drugs, right? Like, we need to ensure that we have an appropriate study design that can really robustly evaluate efficacy. But if there are potential signs of efficacy, how can we reduce placebo, time, which is critical for participants in a study, right? So I think that's one challenge that all of us face in our trial design. So are there potential options like crossover design, you know, where at a certain time period, then everybody then receives active study drug, and maybe that's when, you know, 12 months out from that time point, you are able to evaluate survival, comparing the impact of an early start treatment intervention versus a delayed start treatment intervention. So yeah, those are things that I think keep us up at night, just sort of thinking about what makes the most sense.
And one of the ways that, you know, potentially could be helpful and simply utilized is interim analysis. When you have like 50% of the data, 75% of the data, yes, it eats in to sort of your alpha, if you will, to some extent, but it allows you to have that opportunity to and the double-blind portion a little faster if you're seeing that positive result instead of going through that entire time period. So agree, it's a fine line.
Thank you, everyone. That was so interesting. And kind of tagging onto this discussion, could you expand on what challenges you encountered, or what you think the challenge, like what challenges exist to set up a phase 3 trial in your case or generally? I know it's quite a lot of challenges, but maybe like the ones that you found most difficult, or like the major ones.
I think maybe I can start. As a small company, funding is one of the biggest challenges that we face, right? These are very large global programs, and finding the from the banking world and the VC world, or investors in general, the amount of money that is needed to conduct the phase 3 programs is probably by and large the hardest challenge that we face at Clean. In addition to that, when you're doing specifically for CNMAU8 and Restore a long trial, and having those who have ALS buy into the program for the duration. We have, as I mentioned earlier, we have the interim analysis that hopefully makes it more appetizing for folks to stay on therapy for a longer period of time, but also having virtual visits as an option as they progress through the program. So it's not an onerous trial, but having folks enter a program and stay on therapy. I think different parts of the world and different regions view and the dynamics of being in a trial is very different. In the US, as an example, folks like to get on a program and have it move fast and move on to the next item that they can try in terms of their arsenal that's available. Outside of the US, being on longer programs is much more accepted. So one of the other challenges that we are going to face as we start the program is having folks enroll and stay in the program. Those would be
Thank you, Martin.
Would be the biggest challenges for us.
Thanks. And Lubia, you have your hand up.
Yeah, I mean, I think that we have several challenges, scientific, from a scientific point of view and from the operational point of view. Of course, you want to have the right population. So I think that's the very first challenge. You have to choose the right population that you'll see more benefit, or that will be more evident. So then you can translate that to a broader population. Then, of course, to select what will be excluded, what conditions, which it's never pleasant, because of course we want to help all the patients, but that's not possible. And then, of course, to have the sites and as much as you can representation, globally in talking about a phase 3 global study, and to have those sites that have access to the patients that you are looking for, right? Because sometimes they do have long list of patients, but then that specific kind of population, early patients or whatever, that might be a little bit more difficult to find with, Didrik.
Yeah, thanks, Lubia. I mean, I agree with everything what's being said, but I've also worked in other fields. So I would like to flip it a bit and actually talk about the things that are very well organized within the ALS field. So, for example, this forum today is great for companies like us to be able to present our clinical trial plans and raise the awareness in the community. So I think that is one challenge that is now all of a sudden getting lower. I think the organizations like NEALS and TRICALS that are supporting the industry in setting up the correct trials and be able to find the right sites is also something that is very well organized within this field. and just educating the community about the possibility of participating in clinical trials is very well in the ALS field, and that helps us in generating awareness for our studies and getting participants interested for our trials. So those are all upsides, maybe, compared to the downsides that were mentioned before.
Yeah, thank you so much for that, Elisabetta.
Yes, thank you. In Italy or in Europe, generally speaking, but in Italy specifically, one difficulty is the bureaucracy. So we know that there's an harmonized process across Europe, and the agency have to respect the time, the scheduled time, but we lost a lot of time in order to receive the first approval of the study, because there's a lot of requests from them, not because we didn't write the study properly, but because they would like to receive additional information, and the process to revise the new information that we provide requests a lot of time from their side. And this is a very big issue, because we received money in order to complete a trial in at least 3 years. But if we spend 6 or 7 months to receive the first approval, we have lost a very high amount of time. And this is and also after the first approval, we have to interact with each site in order to define an economical agreement. So sometimes we lost additional 5 or 6 months in order to receive the signature of the agreement. So we have lost 1 year. And so I think that the big issue is the bureaucracy in Europe, but in Italy especially.
Wonderful. Thank you so much. Well, wonderful. Thank you for sharing. It's frustrating. So does anybody else have anything they want to add on top of what's already been said? Otherwise, we can move to the next question.
Okay, I'll bring up the next question here. We've gotten a few questions actually about international participation. I'm really excited to see there's so many sites around the world for these trials. A few additional questions on that. So how would people outside of these designated countries get access to these trials? And are there any opportunities for the Global South to participate more actively in some of these trials? So would love to hear from all of you.
Maybe I can go first. I mean, it's always difficult to find the correct countries and sites for your particular trial. There's always more places to go ultimately than that you can select and that you can manage, because trials are complex and expensive undertakings. So you also don't want to be too broad in all the places where you place your trial. So we have, based on the support we got from Niels and Tricol, selected a certain amount of countries for our particular trial, 13. Cross-border is always possible if we are not in a particular country. Of course, there are language barriers. We also measure patient-reported outcomes, but if those are not an issue, then people from other bordering countries could also participate. And I think ultimately, as we progress the drug through the study, maybe we will also go to more countries in other studies. But we are currently limited to the 13 countries we've selected, which are indeed in North America, Europe, and Israel.
Thank you, Director. Anyone else have comments there?
Yeah, I can refer to the Restore program as well. I think the good news about the Restore program that it is a relatively large study. So we will have a relatively large footprint across the globe, starting in the Asia Pac region, with Australia starting first in 2026 and moving on to Europe and then North America thereafter. We don't have the exact sites, but as Diedrich mentioned, we too are working with the NEALS and TRICALS groups to determine the best sites. But we're looking at approximately 700 patients. So it is a relatively large program, and that's sort of the benefit of this sort of a study that we take.
Thank you, Marjan. Any other thoughts on this question? All right, Martina, do we have any additional questions to address today?
Yes, we have actually a couple of questions that are specific. Specific to compounds. Marjan, thank you so much for typing the answer to those that were directed at you. For Steve, could you expand on why you're only enrolling pre-symptomatic patients in the Atlas study? There's a question from Alper in the Q&A section.
Yeah, that's a great question. Yeah, so within Atlas, we want to evaluate whether early treatment initiation with tofersen in individuals who are pre-symptomatic carrying a SOD1 mutation, so those at risk, genetic risk of developing ALS, whether intervening early upon evidence of ALS disease activity with a neurofilament elevation may delay the time to development of ALS clinically and or slow the decline in function once the disease emerges. So we have set up this trial specifically to look at the earliest possible treatment intervention based on biomarker evidence of disease activity. And within the study, individuals can move to other study parts depending on their neurofilament level, depending on whether they develop clinically manifest ALS. So these individuals who enroll in the study who are pre-symptomatic, who may develop the disease over the course of the study, will then continue to be evaluated in other study parts. And the tofersen program has run multiple studies in symptomatic SOD1 ALS. So we're trying to better understand the timing of treatment intervention on that overall impact on clinical outcomes. So with the VALOR and open label extension data set, we've been able to evaluate the impact of initiating tofersen in a symptomatic SOD1 ALS population, and then able to separate out earlier treatment intervention 6 months earlier for participants who started tofersen in the VALOR study and continued on, compared to those in the placebo group of VALOR who started tofersen 6 months later in the open label extension. So we've had some insights in terms of the impact of earlier treatment intervention in symptomatic SOD1 ALS. And now we want to further extend that by looking even earlier pre-symptomatically. Does earlier pre-symptomatic intervention yield better outcomes? And that's essentially what we're evaluating in Atlas.
Thank you so much. I think we still have a few minutes, so we can do a very quick answer to this last question, which is what do you estimate to be a reasonable timeline for an FDA-approved phase 3 to be available to patients? I know it's a tricky questions and specific to FDA, but you can refer to your jurisdiction if you're not within the FDA jurisdiction. Just to give people, like the trial is only one piece of actual like access to the compound. So it's just to give a picture of what that means.
I mean, I can talk a bit about our timelines for our particular trial. It is a phase 3 pivotal study, so that would mean that if successful, we would move forwards towards registering or trying to register the drug with health authorities. We plan to start first enrollment in the Q1 of next year, so 2026, with an approximate enrollment of about a year. So then you're in 2027, and if you then add about a year of treatment, or that's the first phase of the trial, the double-blind phase, as Lubia explained, you're in the beginning of 2028 when we read out the study results. So that is somewhat the horizon for our particular study. And from there, we would start, or we would engage upfront, of course, with health authorities, but from there, if successful, would follow a possible registration trajectory. So you are well into 2028, roughly.
Thank you, Diederik. That's helpful. Anybody else want to comment or have a different timeline? Please, Marjan.
Thank you. I think, as we've talked about for the phase 3 program for Restore, it is a long program. So the confirmatory phase 3 program will take several years to not only enroll, but complete double-blind and long-term extension. But what's unique for CNMAU8 at the moment, as it pertains to the FDA, is the opportunity for approval while the phase 3 is ongoing. At the moment, if you all recall, and Ben presented in the presentation, during the double-blind phase of the Healy platform trial, we had a positive signal for changes in NFL. And as such, within the Act for ALS Constitution, regulatory language, I should say, we have the ability to utilize this biomarker data and link to clinical efficacy, as we are currently working on at the moment for a potential application in the United States that would be sort of concurrent to our phase 3 confirmatory platform. So for CNMAU8, we do have the opportunity, depending on how the data reads out, for approval in the US if we are able to submit based on this data in 2026. And then end of year would be when we would expect to hear from the FDA in particular, if this pathway is available to us.
Thank you. Steve.
Oh, thank you. Yeah. Just to add on, Qalsody tofersen is in a bit of a unique position within the US. It was approved under the accelerated approval pathway based on a reduction in plasma neurofilament light chain levels. And under this pathway, the FDA may approve therapies for serious conditions where there is an unmet medical need, and a therapy has been shown to have an effect on a surrogate endpoint that is reasonably likely to predict clinical benefit. So that was the pathway within the US. And as part of that pathway, in order to confirm clinical benefit, that's why we're continuing to conduct the ATLAS study. And then beyond the US, Qalsody's been approved in 41 countries, I think, at this stage, based on that last slide. And we continue to evaluate additional regulatory approval expansion. So,
Steve, just to expand on that, 41 countries where Qalsody is approved, where can people find information about whether their country has the approval?
That's a great question. I might have to get back to you on that because I'm not sure if there's a one-stop website that provides that information. But yes, I can certainly look at that and send it to this group after the call.
Wonderful.
Thank you. And if I can step Yeah, if I can step in just for a second, we have a briefing note on our website on tofersen as well as other compounds, and we try and keep the approved list of countries, so the list of countries where it has been approved up to date. It might not be completely up to date, so Steve, I'll actually invite you to take a look and let us know if it's not. I know you've listed some of the countries on the slide, so I'll double check with that. But it's a place to start, and then of course let us know if there is a official Biogen list somewhere.
Yeah. Thank you so much, Martina. You saved me there. So yeah, that one slide does provide the current summary, but yeah, we can certainly work together to update your site as well.
Thank you. I think if there are no more people that want to comment on this specific topic, I'm gonna hand it back to Jess to conclude this wonderful webinar.
Well, I'll start by saying thank you. I just want to take a moment to express our gratitude to all of you for making today's annual clinical trials update possible. On behalf of the International Alliance, a sincere thank you to our presenters, our panelists, our moderators for such an informative and engaging session. We truly appreciate your time, your expertise, and perspective you shared with us today. And of course, to all of our participants joining us from around the world, we're grateful for your engagement and thoughtful questions throughout the webinar. I'll just wrap it up. I think someone said at the beginning that they wanted to say this. I'm going to say it for them. I want to wish you all a wonderful morning, afternoon, and evening, wherever you may be in the world. Thank you very, very much.